Thank you so much. It's a real honor to speak to all of you today. I have a provocative title to my talk. I've called it Genomics in the Post-Genome Era. And I have a very particular meaning when I talk about this. I grew up in the, I would say, the genomics era, but I missed it. I haven't sequenced many genomes. My laboratory doesn't do a lot of sequencing. But what we do is use the information that's been made available by genomics and try to convert that into human therapies. So we sit on the shoulders of giants, essentially. But we don't actively explore. There was a time, especially in oncology, cancer, which is the discipline that I practice, there was a time where there was a kind of sobering moment in oncology when the human genome was sequenced, and following that, cancer genomes began to be sequenced. The pre-sobering moment, the hype, was that the sequencing of these genomes would immediately reveal a whole bunch of therapies for cancers, and in fact reveal the mechanisms by which cancers proliferated, grew, and metastasized. And then came the disappointment, which was that the genome didn't reveal a lot of information. It revealed some information, but not a lot of information, especially in the therapeutic front. And that disappointment continues today to some extent. So, for instance, when the disease that I treat, mostly lymphomas, leukemias, especially acute myelogenous leukemia, the genes that are involved in the mutated genes found in AML are genes that have to do with methylation, RNA splicing, things that you wouldn't imagine. You wouldn't imagine at the first glance would immediately tell you why these genes are involved in that particular kind of blood cancer. And yet they are. We know clearly they are. And in fact, it's an ongoing investigation in my lab and other laboratories to figure out how these genes that seem to be unrelated at the first glance, they're not, for instance, genes that regulate the cell cycle. They're not genes that regulate at the first glance, the typical proliferative capacities of a cell. But somehow or the other, there's a message that we're missing in cellular biology that was not solved. So there was a disappointment that came with that. But now we're realizing that we, as I said, in this, what I would call the post-genome era, we're finding new ways to treat and think about cancer in particular, which I think are profound and interesting, and they all rely on the information released by the sequencing of genomes. So I'll focus on that. I'll focus on physiology and focus on cancer therapeutics. So I suppose that the central theme of my talk is this, which is that the news of my death has been vastly exaggerated, sometimes attributed to Twain. The news of the death of the genome has been vastly exaggerated. This disappointment is actually disproportional to the actual things that we're doing now with the genetic and genomic information that we have from cancer. So I have 4 stories that I'll tell you. My lab works on many other pieces, which I won't talk about today, splicing, among other things. I talked to Phil about it just now. But let me start with this one story, which I think I like to tell stories. So let me start with this one story. So this is about immune therapy for blood cancers. And by the way, this data is being revealed publicly right now. So as in like this minute. And this is not a joke. So I'll tell you exactly what. But the conception of this project goes back many, many years. This is a house that my family goes to in Oaxaca in the winter. It's built on the water, as you see. I call it the most unsafe house in the universe because that is a pool. It's 50 feet above the cliff, and it has no railings. You can only make this. It's a very important house architecturally. It belongs to an artist, Gabriel Orozco, a great close friend. And it is actually built after, the model is after an Indian observatory. So that pool that you see on top, which is an inverted globe, in India was the inverted globe of the cosmos. It was used in the 15th and 16th century to have an inverted shape of the cosmos so that you can figure out where the stars and the horizon was moving, because the horizon is, of course, an upside-down globe as well. But the idea, so I was there about maybe 6 winters ago, 4 winters ago, 5 winters ago, sitting at this house and thinking about inversions. And I was making this drawing. I still have this. This actually happens to be the logo of one of the companies that I've started. I still have this. I was making these inverted drawings, so you can make, so this was, I was drawing seabirds. And you can make an upside-down drawing in which you take a stencil almost, and you can spray paint with black paint on the white stencil, and you all of a sudden get a bizarre and peculiar inverted drawing. But I was also thinking about how we might be able to treat and cure a very lethal cancer, which is really, whose needle has not moved in the last 50, 60 odd years, and that's acute myeloid leukemia, especially relapsed refractory acute myeloid leukemia, where the mortality rate rivals any of the worst cancers that you can imagine, including pancreatic and other forms of extraordinarily lethal solid tumors. The problem is that hematopoietic stem cells and progenitor cells, the cells that give rise to the myeloid cells in our body, the lymphoid cells in our body, but importantly, the myeloid cells in our body, neutrophils, macrophages, red blood cells, platelets, et cetera, and AML cells share common antigens. And to my mind, until now, for AML, and not for other diseases, we have never found a unique tumor antigen that we can target. I'll talk about why that's different from another disease that I work on, ALL, acute lymphoblastic leukemia. AML, we haven't had a unique tumor antigen. Every time we've targeted anything in a acute myelogenous leukemia cell, it's turned out to have its counterpart in some myeloid cell. And therefore, you get dose-limiting toxicities constantly. And you reach a ceiling, you reach a limit, and then you can't go any further, because the more you treat the patient, the more toxicities you accumulate. And so that ceiling is almost a natural ceiling. It's been true for virtually every drug that's been tested, every small molecule, every targeted therapy that's been tested in this space. And so in some ways, we've really not moved the needle for this particular cancer. So the solution to this problem, again, going back to this theme of thinking upside down universes, upside down models, upside down pictures, was to ask the question, what if we, I'm trying to advance this cursor, what if we imagined that, again, let's imagine, this is again, these drawings, these actually original drawings from that session that I had 6 years ago or 7 years ago. What if you imagined a cell where you had a normal cell and a cancer cell, and they happen to share the same antigen? Now, for decades, we've been monomaniacally focused on the cancer cell, trying to do something to the cancer cell, trying to kill it, trying to change it, trying to make manipulations of the cancer cell. But the inversion of that, and so in this case, I've put up an antigen. In this case, it's CD33. I'll talk about that in a second. The problem is that you can target CD33. There are many, many different antibodies, CAR T cells, et cetera, that target CD33. The problem is that they kill the cancer cell, but they'll also kill normal myeloid cells. And then you're stuck, like I said before. But the inversion is the following, which is that instead of targeting the normal cell, by that time, by 6 years ago, we had figured out tools by which we could eliminate the antigen on the normal cells. And now, all of a sudden, you've bioengineered a unique tumor antigen, because these patients will often receive transplants. You can manipulate the donor graft, and I'll show you a picture of that in a second. You essentially eliminate the antigen from the normal cells, thereby essentially allowing this process to happen, which is that if you, again, this cursor is working. If you now target because you've engineered a unique antigen, you can use CAR T cells, you can use an antibody-directed therapy to now kill the cancer cells because they still possess the antigen, whereas you've made the normal cell resistant to the antigen. So in other words, you're using the tumor antigen to stand out like a sore thumb. What does that mean in real life? Well, so this is a patient. On the right side is the donor, and the left side is the patient, or depending on which side you're looking at, patient and donor. So this is the patient has acute myelogenous leukemia, pointed by the arrow. And they're going to receive a donor graft. This is a standard. They're receiving standard hematopoietic stem cell transplantation. But now what we do is, so these are the stem progenitor cells from the donor, and they're matched to the patient. What we're going to do is we're going to use CRISPR to delete out CD33. So now you get a CRISPRed CD33-less hematopoietic stem progenitor cell. Those lines indicate that it cannot be attacked by a CAR T cell. These are all in principle. I'll start showing you data in a second. You can't attack it with a CAR T cell or an antibody toxin. So it's immune to the CAR T cell and the antibody toxin. And then now you give the antibody toxin. That's the AML cell, the cancer cell, which is now sensitive to the antibody toxin. And in fact, you can do one more thing, which is you can also take a T cell, a CAR T cell from the donor. Remember, the patient has now become hematopoietically reconstituted with the donor cells. So you can take a CAR T cell from the donor. It actually comes out from the same leukapheresis as you do. And now you can attack the cancer cell. Is that concept clear to people? OK. So we published a paper on this, and we patented this technology. There are several groups that also did it, but we happened to do it. This is how science works. We happened to do it 2 weeks before all the other patents were filed. It's made a big difference in my life. But this is the first experiment that we did, in which we put in AML and a leukemic cell line into a mouse. We treated either with, and again, we deleted the CD33. This is a mouse experiment. This is an immunocompromised mouse, a non-skip gamma mouse. We put in human leukemia. We've done this experiment with primary human leukemias. We've done this with cell lines. We've done this at various combinations. We'll talk about some of that. And then you treat with either the antibody toxin, which is called Myelotarg, gemtuzumab ozogamicin. That's why it's called GO in these slides, or the CAR T, or a combination. And what happens is that, as you can imagine, if you don't do anything, the leukemia grows, the mice die. But if you put in your, sorry, I'm going to go back. But if you put in your, either the antibody toxin or the CAR T cells, the mice essentially are cured. And we've done this experiment in various combinations. Again, I'll point out on the right side of the slide, you see the dead mice. Those are the mice in which obviously the leukemia grew and took over the bone marrow. On the right side of the slide, you see either the combination of either the CAR T or the CAR T plus gemtuzumab ozogamicin, or GO. What's interesting about it is that when you use CAR Ts, and uniquely when you use CAR Ts, you get a dip in the hematopoietic reconstitution, which worried me enormously initially, but then you get a rise again. And we still haven't quite figured out what that's because of. We think it's because of intramedullary, so in the bone marrow, the lysis of tumor cells creating an inflammatory microenvironment, which doesn't allow the hematopoietic engraftment to happen as quickly as just treating with an antibody toxin. We're not sure about that yet. That's something that we're exploring. It'll become very important in a second. And this, of course, is the picture of the animals that were cured with this treatment. So in 2018, we issued a patent. We created a company, and I'll talk about why that's important, for at least for my ecosystem. I cannot do both the preclinical and the clinical trials at the same time. And that's extraordinarily important. I'll come back to this idea that there's an ecosystem that's necessary to move a human medicine from the laboratory to becoming a human medicine. And that ecosystem is extraordinarily important. And that ecosystem must involve responsible, thoughtful biotech and pharmaceutical companies. And I'm using the word responsible and thoughtful very purposefully, because if they're irresponsible and not thoughtful, it creates all sorts of problems. But if they are responsible and thoughtful, it creates the right ecosystem for the movement of a medicine into a company. We filed multiple patents following that, IPs. We in-licensed the CAR-T 33, the CD33 CAR-T from the National Cancer Institute. We filed now, I think, 6 INDs, but these were the first. And the first in human studies. This is actually an old slide. I just got permission to release the first human data from this study as of this morning. So we created a company called VOR, which is just down the road, created a GMP facility which manufactures these manipulated hematopoietic stem cells. You can imagine that there's a long pipeline. You can do many things with this kind of platform. I won't talk about all of them. We can talk about them at the Q&A session. And this is the study. I won't go through all the details of the study, but basically, it looks like the mouse study. You engraft with the manipulated hematopoietic stem cells, and please don't go through all the minutiae of this slide. You engraft the CD33 manipulated hematopoietic stem cells, and then you, obviously, if there's residual leukemia left over, this unmanipulated, it will still express the antigen. And we had the big advantage that we have not a great drug, but a good enough drug, which is Mylotarg, which is CD33 conjugated to a toxin, which had now been brought back from the reservoir of the dead because it had accumulated so many toxicities. As you can imagine, if you give it to unmanipulated cells, your neutrophil counts and your blood counts collapse. And so there had never been a successful drug, but because of some patient advocacy, it had been brought back. And that turned out to be very important because it's very difficult. We'll come to this as a question and a problem. It's very difficult to combine 2 new drugs into the same trial. It's very difficult to combine a new antibody or a new CAR-T and a new manipulation of a stem cell into the same trial, because the FDA will want them to be separated out so that you don't have confounding effects. By chance, really by luck and circumstance, we had a drug. It wasn't the best drug in the world, but it was a drug. So we could use just the manipulated stem cells as a drug product and then add the antibody toxin. Am I being totally clear by now? Or have I lost people? Okay. So this is fresh off the press, the first patient. So this patient has extraordinarily nasty AML. I can't even begin to tell you how terrible it is. You can look at the karyotype. It doesn't even resemble a normal cell. It's got deletions, translocations, extraordinarily nasty. It's a P53 mutated AML, so the prognosis of these patients is very, very poor. DNMT3A is a gene involved in methylation. KDM6A is another negative prognostic feature so these patients usually have a lifespan of post-transplant relapse of nearly 90%, and a post-transplant lifespan of about 6 months or 4 months, because they will relapse. So again, I don't want you to focus on what happened, what the patient went through, but they went through standard induction therapy, consolidation therapy, which the standard is going on for 20 years, the old therapy, as expected, relapsed. Then they got what's called salvage, venetoclax, which is not a great drug, but at least keeps them in a salvage mode. And then they had 1.8% MRD positivity. So even after all of this, they were minimal residual disease positive. So that's MRD at 1.8%. So in other words, their chance of dying without a hematopoietic stem cell transplantation was 100%, because there's nothing that will keep this MRD at 1.8%. It will climb and climb and climb, and they will die. So they then got a 10 out of 10 match. This patient happened to have a match, very good match. Then they got standard conditioning with busulfan and fludarabine. And then they got this TREM cell infusion. We haven't got the name of the drug product yet, but that is the CD33 deleted cells. That's the dose. The gene editing efficiency that we allowed for the release was greater than 50%. This person had 88%, so 90% of their stem cells were edited. And of course, this is a purified CD34 product. Any questions so far? Everyone's following me? Okay. So this is the early data from this patient. So the median engraftment would have been at day 11. In fact, this patient engrafted with the CD33 negative cells at day 12. So really no difference. That was the last dose of GCSF. And the important data is what you see right here, which is that rather than their neutrophils and their platelets and their cells collapsing, which it would do, as we gave them, their platelets started to recover. And then this is what happened. So their donor chimerism was nearly 100%. Their CD33 negative cells was, at day 60, was 96, 94, 93%. But the most important thing is that they have now gotten 3 doses of the antibody toxin, and their stem cell, their recovery has continued absolutely on the normal trajectory. So in other words, they have been completely immune. They have become completely immune to the toxin. So this is the natural. This is the ODAC committee natural. So they got 0.5 milligrams per meter squared of Mylotarg at that, and now they're moving on to the next dose escalation, which will be 1 milligram. At that dose, you should expect to see 100% saturation of the receptors, but this is in a patient who's replete. This patient, remember, doesn't have any sink. Their only sink for this toxin is their cancer cells. Normally, you would expect that in 8 out of 9 patients within 14 days, there would have been a drop in their neutrophil counts. But this patient now actually has, in fact, increased their neutrophil counts. At day 20, their ANC has normalized to 3.25, platelets now at 94, and most importantly, no hepatotoxic events. Tiny, tiny, tiny study. We're recruiting 6 more patients this week, and the FDA has asked us to move them by 2 weeks, 2 weeks, and 2 weeks. So by December, we'll have a full cohort data set, but we're obviously very excited about this because it, as I said, proves an incredibly important principle in this platform. And there are many other potential things that can be done with this platform. Well, the second thing that can be done with this platform is that often cancer cells you need dual therapy, so you need 2 targets. And in fact, there are 2 other targets that are present. Gene called CLEC12A, mutated in AML, another gene called CD123. The reason we pick these genes, by the way, is that we have the so-called NOMAD database. And the NOMAD database is a very important database. It's a database of all natural deletions that are found in the human population. And just by chance, it turns out that there are many people who have naturally deleted CD33, arguing that you don't require CD33 for normal function. So that's one of our major screens that we use to pick out these genes. They have to be present in the leukemia and in the normal stem cells. And it's much, much more reassuring to us and to the FDA if they are naturally deleted in some population of humans. CD33 happens to be one gene. There are many other such genes, about 5 or 6 others. I can't tell you all of them, but I can just tell you some of them. So we've done double deletions as well. So we've done a dual deletion. And again, the idea would be to dually delete 2 genes now from the hematopoietic stem cell product. In this case, we've done it with CLL1 or CLEC12A and CD33. It's very efficient. So you get almost 90–95% of deletion. And we've, in the laboratory, treated, again, with the same principle, treated now with dual CD33 and CLL1 antibodies. We have created a proprietary antibody against CLL1, attached it with a toxin, and again, you get the same results. You get, in the mouse at least, you get full engraftment with every cell type. The leukemia still expresses CLL1, expresses CD33, and when you treat, you can cure the animals. The problem with dual targeting, as you know, is that it runs the risk of translocations. So the FDA told us, wait a second, you can't do that. You can't do a double targeting. We've argued back with them, saying that this is a totally lethal disease, and this argument continues. As I said, some of this early data from patient 1 will help this argument, but we appreciate and understand that. So we've turned to doing two things, which are interesting. Both of which I'll go through very quickly because I want to get to the other parts of the presentation. So one way you can do this is you can do it sequentially. So you can do one, you can edit one or delete one, wait until the Cas9 is cleared, then delete the second. Now, that's a little bit of playing with fire, although we've now adjusted the conditions. And the reason is that the more days you keep stem cells, human stem cells, in culture, which is how you're deleting all of this from the donor population, the more chance you have of the stem cell population decreasing. But we've managed to find what I call the Goldilocks spot, so we can do double editing. We've also done base editing. Now, base editing, remember, technology that came out of Boston, involves changing bases without making a double-stranded break in DNA. So I'll talk quickly about base editing. We've done both sequential and base editing in animal models. Again, we have showed the same thing, that you can do a double edit. You get equivalent engraftment, no difference between the unedited versus edited cells if you treat them equivalently. And so, and in the AML models, again, same data positive. I'll go through the base editing data very, very quickly because I'm short on time. But essentially, you can get very effective dual base editing. Here is a example of the double base edit. And you get, we've shown that, at least the cells remain functional using standard assays like phagocytosis, cell motility, challenges with E. coli, and so forth. These are things that the FDA asked us to do. This is now a hematopoietic stem cell that is lacking both CD33 and CLL1. And if you, again, treat with the antibody toxin, you get very strong, at the bottom, you get in the dual edited cells, you get very strong cell killing, whereas in the unedited cells, you don't get cell killing. There are some off-target effects. On target, of course, predominates with CD33. And then there's off-target effects, which I think are within reasonable limits, at least in genes that don't seem to affect their function as judged by animal experiments. So I'll move on to the second piece of this. But before I do that, let me just pause for 1 second because this is a modular talk. So are there any questions that have directly to do with this? We'll have a question and answer session. Has any questions directly to do with this part of the talk? Yes?
Congrats on amazing data.
Yeah.
Is there any evidence that the enriched gene-edited cells
Yes.
in the patient also harbor additional mutations due to off-targets?
We haven't tested that yet. The only thing I can tell you is we got 100% chimerism, so it's easy to test. So we will do genomics for once in my lifetime on real patient data. Yes. Any other questions on this first part? Nope. We're good? OK. So
I have a quick question over here
sorry.
on the right.
Yes, sorry.
Your left. Manoli Skelos from MIT. So my question is about CD33. Basically, when you're actually targeting these cells, where in the body does it go? Because CD33 is also a marker of microglia in the brain.
So important question. So there are two concerns about CD33. One concern was that there would be toxicity to Kupffer cells in the liver, which are macrophages, resident macrophages in the liver, and microglia in the brain. Now, we know that that's not going to be the case with Mylotarg. at least used in the on-label use, because it's been used for decades. So you don't get brain toxicity or you don't get neurotoxicity, and you do get hepatotoxicity, but at doses that are much, much higher, 10-fold higher. So at 5 mg per meter squared and higher doses, you start getting hepatotoxicity. We're going to stay in the 1 to 2 range, and the highest range is 3. And so far, we haven't seen any hepato or neurotoxicity. Now, with the CAR T cells, it's a different story, because those cells do traffic in the brain. So that study will be a different study, and we have to be very careful about both the Kupffer cells in the liver and the CAR T cells in the brain, both of which we will be able to assess once these patients live longer and longer.
Got it. Thank you very much.
Thanks.
Beautiful work.
OK. This is a collaboration that I did with Ron Vale. This is also a study that's recruiting. I'm only going to talk about clinical studies today, but also really about the transition from the laboratory to a clinical study. So this collaboration is the idea of using a monocyte as an effector cell. So remember that CAR T cells have been used for a long time, genetically modified T cells, CAR T33 in our case, and other CAR Ts. I'll talk about those in a second. But the question was, why don't we use monocytes? The problem with monocytes, as you know, is that they're part of the innate immune system, and they don't have an adaptive capacity, so they're not specific for any antigen or cancer. So we said, well, what if we made them specific for an antigen or a cancer? So just the background, myeloid cells are the most abundant cells in solid tumors. They have tumoricidal activity. We know that from in vitro and in vivo experiments, but they don't have specificity, right? So the question was, how can you impart specificity to a myeloid cell? So we created what is called what we call a CARP, P for phagocytosis, work done in Sargil's lab, and Carl June, I don't know if Carl's here, but have shown similar data as well. But we've done work in parallel. So just to create an analogy between a T cell, so in a CAR T cell, you have, let's say, CD19 and its cognate receptor on the other side, which is usually a combination of the CD19 in the extracellular domain, or anti-CD19, CD3 zeta, 41BB, or CD28, various activators in the intracellular domain. So everyone, I assume, is familiar with that. We created a CARP receptor, which again, in this case, has CD19. This is not going to be our first product, but this is a proof of principle. CD19 on the extracellular domain, so we'll target B cells or B cell malignancies, and an engulfment receptor. And there are many variations of engulfment receptors. But most importantly, we've used limb domain engulfment receptors. We've used many variations. But one thing that's important is that these engulfment receptors work very well. So this is this was one of the first in vitro videos of this CARP now swallowing up a lymphoid cell, CD19 positive Raji cell. I like this video because it shows you actually two things that are very interesting. Number one is you see the engulfment, and the number two is that you see that once the cell is engulfed, what happens to it is that it actually bursts inside the macrophage or monocyte and releases all its contents into the macrophage or monocyte. That turns out to be important because that is a potential source of neoantigens being cross-presented now to T cells, and I'll show you data for that. So our first version was a very simple CD19 on one side and the engulfment receptor on the other side. We added a PI3 kinase co-stimulatory domain to this based on work that had been done in my lab and in Ron Vale's lab that showed that if you stimulate the PI3 kinase using a stimulatory domain, you now get about 8 to 10-fold enhancement in the capacity to phagocytose the cells. We've since then added even further co-stimulatory domains to this to raise the phagocytic capacity of these cells up to now about 20-fold from just the old version 1 receptors. I can't tell you what those are because they're still being in patent process. I'm sorry about that. So we've started 3 to 4 studies on this. One of them is in CD5 positive T cell lymphomas. The logic there is quite obvious. You can't use a T cell to kill a T cell because it will eat itself up, whatever antigen is you're directing it against, if it's present on the normal T cell. So, but you can potentially use a myeloid cell to kill a T cell. So it's directed against T cells. We have one against, for ovarian cancer. We've just opened a study for TROP2 positive breast cancer, breast and ovarian cancer. So in fact, that study is recruiting more actively than the ovarian cancer that's coming afterwards. Again, I can't show you the top-line data for that because we don't have it yet. This is the mouse study. This was using a melanoma antigen, and this is this is a melanoma model. We call these ATAK cells, attack cells. I can tell you later about how we manufacture them and how they're made. But basically, in melanoma, you see a very clear signal for transplanted attack cells. These are transplanted at 2 × 10⁶. You give them 8 infusions. They're quite easy to harvest both in mouse and humans. They accumulate very nicely in tumors, and they have very strong tumor control properties, at least in the mouse model. I won't show you the data that I have because I'm moving through this quite quickly. I won't show you that we see very good evidence of cross-presentation. So we've used a tumor that has ovalbumin in it, so an ova mouse, and you can see a very striking increase in ova-specific T cells if you give them the GP75 specific monocytes. And that is this illustrated very crudely here, is that the attack monocyte not only has a direct mechanism of killing the cancer cell, in this case using HER2, but also activating, having ingested the cancer cell, cross-presenting to the T cell, which then activates killing of the tumor cell. Some of these studies are still ongoing, so it's a little bit early. So we've dosed 2 patients. They've tolerated the doses. I can't tell you the final top-line data yet because we don't have it yet. I can tell you that one of them is now 10 months post-dosing. So I don't know what that means. All right, any questions on I went through this rather quickly. I want to finish this last piece. So any questions on the attack program? Good with that? Okay. All right, this is probably the proudest thing that my lab has done, and it's not a lab project. We created the first CAR T cells in India. This was a population which had never, CAR Ts were never available. We made a fully integrated licensed CD19 CAR T product, a collaboration that we had with, we developed this with Spain, with Hospital Clinic Barcelona. We bought, basically paid out of our own pockets, bought 2 Prodigy systems and created, using Indian engineering, 1 and now 2 open systems. And by open systems, I mean they don't have anything that is proprietary and belongs to, aside from a Clinimax unit, anything that involves Prodigy or its consumables. Why? Because we want to bring the cost down from about $500,000, about $1 million all in, including all the therapy, et cetera, down to about $40,000, maybe $20,000. Now, that sounds incredibly expensive, but that's about the cost of a bone marrow transplant in India right now. So we wanted to bring it down to comparable numbers. So this is the unit. I'm sorry I'm showing you photographs like this, and this was because we built this unit during COVID. We essentially created a bubble, which was you could only test in and test out. So if you tested negative, you would be put into, I mean, this is a real system, put you into quarantine for 5 days, test you again, and then release you if you left the bubble. But if you stayed within the bubble as the GMP unit was constructed, you could remain open and free. And this is the construction of the GMP unit. I went twice. I made this unit with 2 masks, 16 hours on a flight, 2 N95s. I survived that, which was not pleasant. Then there were the vaccines, which helped my life enormously. But this is a company called Immunil, and I think this is probably the proudest moment of my life, because that bag over there hanging in the first slide is the first patient being dosed. I dosed the first patient, a 10-year-old boy with relapsed refractory ALL, whose mortality would have been 100%, so would be dead by now. So we have now CD19, CD20, BCMA, just filed 2 new INDs for 2 new antigens in lymphoma and myeloma. As I said, we finished GMP facility. We were inspected by the Indian FDA, then by the EMA, and we're waiting for a FDA inspection. They're backlogged. We finished QC in early 22 and have now routinely gotten 40% plus of CAR-Ts given they're pathogen-free. The QC is exactly comparable to the QC that you would see in a CAR-T facility in UPenn, for instance. I know Bruce is on our board. Bruce, I saw him in the morning. Hi, so Bruce, thank you for all your help. So again, fully integrated. We have now started making our own lentivirus. So this is the first GMP lentivirus being produced in India. The titers are totally comparable. We are now down to about $40,000 of COGS. Cost of goods will be down to about $20,000. And please don't disclose this data because this is a private session. So what this allows us to do, of course, is not what I'm particularly proud of, is to treat and cure patients with relapsed refractory ALL and myeloma. It allows us to do a completely novel clinical trial design, which I am most excited about. Because if you bring COGS down by 10-fold or 20 or 30-fold, you can do a completely novel kind of trial in which you say, well, instead of waiting for someone to fail 3 lines of therapy and then give the CD19 or anti-myeloma CAR-T, what if we gave them, because we can afford to, what if we gave them the therapy at first remission? A completely different idea. And I'm very, very interested in this trial because the one thing I've learned in oncology, which I think is, I learned from Bert Vogelstein the first time I met Bert, is Bert told me that there are 3 zeros between a million and a billion. And bizarrely enough, oncologists have forgotten that there are 3 zeros between a million and a billion. The relevance of that is if you have a billion cells that you're treating, it has a billion possibilities of escaping from your therapy. If you have a million cells you're treating, that is reduced by a factor of 3 logs. If you have 1,000 cells you're treating, that is reduced by a factor of 3 logs. We seem to have forgotten this idea, but of course this is the basis of adjuvant therapy or in oncology in general, is that if you treat a slower disease burden, you also get the double advantage not only of treating less cancer and more on-target cancer treatment, but you also get the advantage that you have less chances of relapse and failure of treatment because you have log-fold differences in the capacity of the cells to relapse. The problem is that we can't use this because typically we're using drugs in the 3rd line, 4th line, 6th line, 8th line setting. And by that time, you have that karyotype that I showed you in the AML patient. Every single chromosome is gone, a whack, the mutations galore, et cetera, and of course, you increase the capacity of the tumor to escape. By moving COGS down, you have really the capacity now to do what is quite, I think, a revolutionary kind of study, which is a new trial design. So I'm not going to give you all the data because these are 2 abstracts presented. We have 6 abstracts at ASH. The American Society of Hematology meeting. This trial is called Imagine. 10 patients treated so far of planned 24. Median prior lines of therapy is 2 lines. In the US, it would have been 4 or 5 or 6. 83% for ALL, including some cures at day 90. And for non-Hodgkin's lymphoma, CD19 positive, 77% with an overall response rate of 75%. And this is one example of an NHL patient. So you can see the pre and post. So diffuse, this is a PET scan, obviously. Diffuse adenopathy, actually there's 2 patients. Diffuse adenopathy, which is PET positive all along the legs, very bright lymph nodes also under the armpits. And this is a patient, both patients had a complete response, CR. And this continues into day 90. The ALL patients, the longest has gone up to, I think, day 144. So presumably, hopefully cured. And again, we do use genomics still. That is the rise. You can detect the rise of the T cells using qPCR in their blood. It rises to a peak of several thousand counts, and you get a B-cell aplasia that accompanies this, which is a very good prognostic sign because the tumor cells are not, the CD19 CAR T cells are not just killing the cancer cells, but they're also killing B cells. I talked about this already. I have one last block to talk about. I think I have 6 more minutes. Any questions about immunotherapeutics? The ASH posters will be out in 7 or 8 days, so you can follow that as we go along. I personally have never been more proud than this. The last study that I want to talk about is, again, a study we published with Luke Cantley. It's published in Nature, about 3 years ago. And again, has this same theme, in which we stand on the shoulders of giants using genomics and try to make something new out of these genetic studies. And this has to do with PI3 kinase, of course, Lou's work everyone knows here. This was back when he was in New York, in my city. Unfortunately, it's come back here, but fortunately for you, I suppose. Anyway, this goes back to studies that were done in the mid-2000s in PI3 kinase mutated breast cancer. And the very surprising and disappointing data there was that even though this is a very frequent mutation in breast, endometrial, and other cancers, very specific PI3 kinase alpha and delta inhibitors fail to show clinical responsiveness. And we really scratched our heads about this. I was in one of the trials, the AML and the MDS trial for this. We got very poor, these are the Kaplan-Meier curves from the New England Journal of Medicine study, and this is a comment by Ian Kroc saying that it's a bizarre surprise. And this was the kind of sobering data that I talked about. We have mutations, you have the genome, you have a sequence, you have a drug, and it still doesn't work. And we scratched our heads and really were extraordinarily disappointed about the study, as of course were patients. But then I had lunch with Lou, and Lou's very hard to have lunch with because he doesn't eat anything. But I had lunch with Lou, and we began to, I said to him very casually at lunch, I said, you know what was bizarre about the study as a clinician is that many of the patients who were pre-diabetic became frankly diabetic. So their insulin spiked, their glucose spiked. We had to send them to the endocrinologists. The endocrinologists put them on insulin and manage them on insulin. But if they were pre-diabetic, they became diabetic. If they were non-diabetic, they began to have glucose intolerance. And this was a back of the napkin discussion, which I love having. But this basically shows that same data in the mouse. So if you give PI3 kinase inhibitors in the mouse, you see this similar situation. You see this spike in glucose. So essentially, a week or two after we had this discussion, we went back to the laboratory, dosed mice with, in this case, I think it was copanlisib, which is an alpha and delta specific inhibitor. And you can see that both the insulin rises, insulin on the right side, shown by C-peptide. BKM120 is a PI3 kinase inhibitor, BYL719 is one, and of course is a vehicle control. So we can reproduce this diabetic phenotype in the animal. And then we argued, or we hypothesized, that what was happening is that the inhibition of PI3 kinase, and again, this shows you the blind spots that we have as tumor biologists. We think of the tumor as if it's sitting in space. But of course, it's not sitting in space. It's sitting inside an animal. It's sitting inside a human. And so what happens inside a human is that, yes, the PI3 kinase is inhibiting the inhibitor. The drug is inhibiting the PI3 kinase in the tumor, but it must also have physiological effects because there's a whole animal. It's not just sitting in space. And in fact, the physiological effect is now the release of the body. This is the hypothesis. The body thinks that it's going through starvation because you've inhibited the central detector of nutrients. And the response of the body is essentially your liver starts releasing glucose. That glucose then prompts the pancreas to release insulin. And now the hypothesis was that that insulin reactivates the very genes or the very downstream pathways that the PI3 kinase inhibitor was supposed to inhibit. I liken this to the woodcutter, the famous fable of the woodcutter who sits on the tree and chops off the limb that he or she is sitting on. And in fact, that turned out to be true. The simplest way to see this is the following. This is a tumor imaged by PET, either given vehicle or a PI3 kinase inhibitor, BKM120. And you would imagine, since PET detects the glucose flux into the tumor, you would imagine that, in fact, if the tumor was dying, you would imagine the glucose flux would go down. In fact, just the opposite happens. And we've done this in humans now as well. So in fact, just the opposite happens. In fact, the glucose flux goes up. And what really happens within the tumor is I won't go through a lot of the details, but in summary, what happens is what I just told you is that you get a glucose release into the pancreas. Pancreas then releases insulin. The insulin, now we know, binds to the insulin-like growth factor receptor, and that receptor reactivates multiple genes that are resistance mechanisms to PI3 kinase. We've shown this now in multiple models of cancer, I think 12 models of cancer. And what's interesting is that it's mutation agnostic. It's true for PI3 kinase mutated tumors, but it's also true for PI3 kinase non-muted tumors, including pancreatic cancers in the mouse. I think I have the mouse data here. This is just that same idea repeated, which is to say that the inhibitor certainly inhibits PI3 kinase alpha in the tumor cells. This is in the case of AML, and thereby inhibits AKT, S6, and mTOR in the tumor. But the inhibitor also inhibits in the pancreas and the liver PI3 kinase, thereby releasing insulin, acting through the insulin receptor, and reactivating the downstream genes involved in tumor proliferation, resistance, and growth. There are two strategies by which you can lower insulin in vivo. You can use drugs, or you can use a ketogenic diet. Now remember, this ketogenic diet is an insulin suppressive diet. There are 1,000 variations of ketogenic diets. The sole role here is to suppress insulin. It's not to make ketones. Ketones are only there so that you can live. The sole goal here is to reduce insulin. We've done variations on this theme. We can use SGLT inhibitors or the ketogenic diet. It turns out that actually the diet plus drug combination is the best. This is the lowering of insulin with the ketogenic diet alone. And you can see that if you do this combination therapy, you can see that the insulin receptor is not activated. And there's a lot of biochemistry in the paper that shows that these downstream things from the insulin receptor, including AKT, S6, and mTOR, are no longer activated. This is the mouse data. This is AML. It's true for allografts of pancreatic cancer. It's true for endometrial cancer. These are PDX models. I think we've taken, about 12 such independent models and shown that you get very profound reductions and very profound effects on the tumor if you combine an insulin-lowering strategy with the PI3 kinase alpha inhibitors. I'll reach my conclusions because I'm out of time. So I guess there are vast opportunities to combine cell therapies with genomics and genetics. The rumors that we're not using genomics is not true, although, as I said, we're using them in completely new ways that I think are fascinating to me as a physiologist and as a cell biologist. Obviously, I talked a little bit about combining metabolic therapies with genomics, the possibility of combining cancers, I didn't talk about degenerative disease. And I'll leave you, the one question that I get asked often is, how do I write books and also manage to do research? I certainly don't do the research at the level of most people in this audience. So, but I do like to make medicines. I do like to do translational research. And the way I do it is by creating or delegating responsibilities to people who are extraordinary. And these people who've taken these projects forward in these laboratories in India, sitting through COVID in a bubble, flying across the country when it was extraordinarily risky, nursing the first CAR-T patients when no one knew how to dose T cells, are just really extraordinary people. Giving advice to us basically for nothing, Bruce, Carl, and folks, and I always remember that because Gary Gilliland, who was one of my mentors, and I'll end with this, said, 1,000 B pluses do not make an A plus. If you have A plus people, if you have people you can give the responsibility to, you can recruit 5,000 people, and you'll never get the same result. You have one person, you have one nurse who knows how to detect an early cytokine release syndrome. That is the person who will save the life of a 10-year-old boy getting their first CAR-T therapy. And I think that in some ways is my theme in life, and perhaps is the message of this conference, which is if you recruit A plus people, you don't need to recruit 1,000 anyone else. You just need one to be able to do that. Thank you very much. I'm happy to answer any questions. Should we sit here?
Wonderful. Thank you, Sid, so much for this fantastic presentation. Let me just sit here, maybe. So now we are going to be opening for Q&A.
Do I need to, I'm mic'd already, so.
Wondering, OK, I think we have one question here. I can barely see with the light. One over there as well. So let's start here. Yeah.
OK, thank you. Siddhartha, thank you. That was absolutely wonderful. I wanted to ask you a question about India, philosophically and more broadly. You are rightly proud of what you've done there. But if the theme of this conference is about trying to take cutting-edge science and medicine to help people, let's just summarize it in that context, through accelerating what technology can do, I think it's important that we all realize that there's 8 billion people on the planet today. Only 300 million of them are in this country. And your work in India, as you said, is the first example of someone building cell therapy to treat the 1.3 billion people who live in India, who represent a population that is much closer to the 8 billion people on the planet than the 300 million here. Conventional wisdom in this country is that innovation can only come from the great US. Interesting that you're an Indian American. So there's clearly Indians who can innovate. Just thoughts on one, is there an opportunity to push much more innovation in countries like India at the cutting edge? Two, what do we, as an industry that's dedicated to helping humans, do to shift the dynamic where obviously American-priced therapies, not just in the world of cell therapy, but more broadly, are completely unaffordable to the vast majority of people on the planet?
Yeah, so very important questions. First of all, that conventional wisdom is all nonsense. I can speak as a former Indian citizen. We're sending rockets to the moon just as much as you are. Not that that's a proof of anything, but proves that technology can be moved around. But more importantly, as I pointed out to you, I have become a believer in what I call reverse tech transfer. So for the last 100-odd years, it's not true 100 years ago, 100 years before the last 100 years, technology moved from places like India north to the global north. And then, of course, in the post-colonial and colonial context, that technology started moving the other way around, coming to places like India. And India is only one example. China is, of course, another very, very large example of this. I'm absolutely confident that during my lifetime, we will see reverse tech transfer. We're already going to see it in the next, I would say, 9 to 10 months. We will launch our first study of BCMA in the after first remission. That study has already been approved. This is for multiple myeloma, again, to talk about a completely new shifted paradigm. We have 2 new INDs that we're filing for new proprietary products developed entirely in collaboration with Spain and with the US, but they're Indian products for lymphoma and myeloma. And then we'll do another study in first remission leukemia, ALL, which will also be in first remission. We're likely to transfer VOR's capacity, not after, again, 3 lines or 4 lines of chemotherapy, but upon first remission. All of these are going to be completely pivotal trials that will change the landscape, I think, of the treatment of at least blood tumors, blood cancers. We will eventually move on to non-blood cancers. Ovarian cancer is on our line of sight. So that's the answer to the first question. The second question is also important. How do we afford all of this? Certainly can't afford $1 million. We can't even afford $500,000. So the COGS has to be brought down by 10-fold, if not 30 to 40-fold. Is that doable? Yes, because we're not making gold. We're not mining. We're taking T cells from a body, making viruses that can deliver receptors to those T cells or myeloid cells, and then injecting them back into the human body. These are engineering problems, and you've seen the famous curves in which in chip technology, for instance, or in other forms of technologies, the prices have gone down 10-fold, 20-fold, 40-fold, 80-fold, 100-fold. Data management is another example. So I think it's very, very doable. You just have to concentrate on the engineering. And there are plenty of very good engineers around, both biological and outside the world of mechanical engineering. So I'm pretty optimistic about it. I think Sadiq had a question.
Sadiq?
Yeah, Sid, really wonderful talk, and the VOR data, obviously, personal highlight to see that patient data actually being translated. So related to India, you mentioned you used the Miltini Prodigy system, and then you made some modifications to make it an open system. I was wondering if you could elaborate on, the hardware modifications that you needed to implement in India, and the QC release testing. That seems to be the biggest challenge with this decentralized type of testing model. How were you able to overcome some of those issues? Did you have to invent new methods, repurpose existing technologies?
Yeah, so some of it is proprietary, so I can't totally give you a full, but we made them. So I made them in collaboration with the Indian engineers, so that now we are basically down to just having the Clinimax. So everything is out of the Prodigy. So everything, we opened up the Prodigy, took out all the bottles, and took out all the pipes, et cetera, et cetera. Our goal is to do the first study with the open system. It's not been in study yet, but what we're finding in both animal POC models and in vitro flow cytometry, et cetera, that the QC has not been changed at all. So we're still getting the same level of transduction, the same QC criteria, the same number of, the same MOI is being used for the Lentivirus. I think that it's possible to do it. Again, not at total liberty to discuss how we changed all the piping, but the open system works really well. And the point is now to potentially even remove the Clinimax completely, bringing down the COGS down to the levels that we talked about, $20,000 to $30,000.
And actually, related with the theme, how are you planning or thinking on expanding on capabilities and throughput to make sure that you can reach to all the patient population?
It's a big challenge. We're doing a couple of things. One of them is there's a lot of training involved. So we're training about 200, the center is now training about 200 nurses to take care of these patients. And we are just, so the Indian FDA, the DCGI, the Drug Controller General of India, asked us to do a multi-center study to show that, it's not just, you just didn't make up this really fancy center in Bangalore, which is the tech hub. Bangalore is a very strange city. You pass through, to get from Delhi to Bangalore is a 3-hour flight, and to get from Bangalore to Electronic City, which is where India's Silicon Valley is a 3-hour drive. And you pass through roads that have been half clogged by garbage. And then you go into a GMP facility, where there is, not one single, we're testing constantly to make sure there's not one single pathogen around. It's just a kind of mad experience. Anyway, but scalability is a big question. We're training, so there's human labor. And the second way that we're doing this is, as I said, we are now beginning to manufacture open systems so that we can handle tens of patients per center per month, up to about 50s of patients per center per month. And it's all about engineering, because it has to do with, again, by opening up the system, you can supply all of these. There's no supply chain issue. You don't have to buy 8 prodigies. We're actually in collaboration, thinking of a collaboration with an Israeli company, which has also done a lot of this work. And so we hope to be able to treat 50s of patients, maybe hundreds of patients per center per month. And the demand is infinite, by the way. If you take lymphoma, leukemia, ALL, and myeloma, it is easily potentially possible in the entire South Asia region to treat tens to 50s of patients per month.
Yeah, really exciting. I think we have time perhaps for one last question. Over there.
Yes, sorry, I can't see you.
Sid, very nice presentation. I think the 5 examples you talked about, I want to think about how do you look at a problem differently. You talk about innovation. You gave the example of PI3K inhibitors. For people in the audience, every pharma company used to have like PI3K inhibitors. They were all doing exactly the same thing. 10, 20 years, hundreds of millions of dollars, maybe billion, nothing came up. So as we are sitting here and looking at genomes to cure, what do you think, we should look at the problem and not follow the same path?
Well, I think, as I said, I mean, the important thing that I wanted to emphasize through this talk is that the genome is not physiology. There's a layer of physiology that's missing. If you want to translate genomic insights into cancer therapies or to human therapies, you need to focus or understand the physiology of cancer in situ. And there's a big missing link there. I think that's the missing link that there's a line in my book, which is that sequencing is seduction. It's very seductive technology because you open up a tumor and you have lots of information, data about which genes are mutated, what genes are translocated, et cetera, et cetera. It's very seductive information. The problem is that is not physiology. Physiology requires a different kind of thinking. It requires thinking in a holistic manner, in a way that understands what the tumor is, if you're talking about cancer, other diseases as well, but it requires a different kind of thinking. And I think that is an incredibly important missing piece in our ecosystem around therapies today. We think about a kind of a linear way of going from genomics and genetics to therapies. The thinking is often nonlinear because we discount features in the animal, in humans, and in cancers that might be resistance mechanisms, that might be countervailing mechanisms, homeostasis, the basics of life we discount and therefore are unable to discover why things are the way they are. Just to finish up, just having this conversation with Phil, we were having this conversation in New York. We still don't understand, as I started off in the beginning of the talk, we still don't understand why certain mutations, for instance, RNA splicing mutations, are so commonly present in leukemias and in some other cancers, breast cancer being one of them. We still don't know. What does RNA splicing, of all things, have to do, a very generalized process, have to do with cancer. And Phil pointed out, and this was at another talk, he pointed out maybe we're just using the wrong conditions to grow the cells or to assess why they're causing cancer. Maybe we need to use different kinds of conditions. It could be as simple as that. The bone marrow is not a lump of tissue in your spleen. It's not a lump of tissue on your skin. They're different environments. If you don't reproduce those environments, if you don't reproduce the glucose content, the oxygen content, the insulin content, which is all physiology, by the way, you'll never know or you'll never discover or figure out why certain mutations or certain genomic therapies cause cancer, and secondly, how to potentially find therapies against them. So this is really a plug to think about the post-genomic world using genomics, but think about post-genomic world in a physiological manner.
Well, thank you so much, Sid. Fantastic. Thank you for being here today. Thank you.
Thanks a lot.
Yes, thank you.